Assalamualaikum warahmatullahi wabarakatuh. Today, I want to explain about DNA extraction. In this experiment, you need to isolate some DNA from a human test subject. Why we need human DNA? Scientists isolate DNA for a variety of reasons, some of which include genetic testing, body identification, and analysis of forensic evidence. DNA extraction is typically the first step in a longer laboratory process. DNA extraction is an important part of that process because the DNA first needs to be purified away from proteins and other cellular contaminants. We need cells because that's where the DNA is. Inside almost every cell in our bodies is a nucleus, and inside each nucleus is about 2 meters of DNA. So, where do we begin? First, we need to collect some cells from our test subject. The skin on the inside of our mouth loses thousands of cells every day. These are the cells we are after. Let's get started. The steps you will follow to purify DNA from a chick swab are shown below. First, collect your cells. Second, press cells open to release DNA. Third, separate DNA from proteins and debris. And last, isolate concentrated DNA. Here are the materials and equipment you will need to purify DNA from chick cells. First, drag the swab over to the inside of the test subject's mouth to collect a sample. Next, you will place the swab into an Expandorf tube. Look closely at the swab. Note that it is covered with hundreds of tiny chick cells. Inside, it, inside each chick cell is a nucleus, and inside the nucleus is DNA. The end of the swab must be cut off so you will be able to close the tube. Using the micropipette, add some lysis solution to the tube. Lysis is a Greek word that means to separate. And then place the tube into the warm water bath. The lysis solution you just added contains two important ingredients, detergent and an enzyme called proteinase K. The detergent disrupts the cell membrane and nuclear envelope, causing the cells to burst open and release their DNA. The DNA is still wrapped very tightly around proteins called histones, and the proteinase K cuts apart the histones to free the DNA. The cells have stayed in the warm water bath long enough for the DNA to be freed from the cells, and we have removed the swab from the tube. Add some concentrated salt solution to your tube. The salt causes proteins and other cellular debris to clump together. Place the tube into the centrifuge. In order to balance the centrifuge, a tube containing water is placed opposite your tube. Click the lid to close the centrifuge and turn it on. Inside the centrifuge, the tubes spin around at high speed. The heavy clumps of protein and cellular debris to sink to the bottom of the tube while the strands of DNA remain distributed through the liquid. Use the micropipette to carefully remove the top liquid, which contains DNA, and place it into a clean tube. The proteins and other cellular debris stayed behind. Add some isopropyl alcohol to the tube. Inverting the tube several times mixes the isopropyl alcohol into your DNA solution. Because DNA is not soluble, does not stay dissolved in the isopropyl alcohol. It comes out of solution. You can now see the clump DNA with your naked eye. Place the tube into the centrifuge and click on the lid to close it and turn it on. This time, after the sample spins in the centrifuge, the DNA sinks to the bottom of the tube. Once the liquid is removed and the DNA is allowed to dry, you can redissolve it in the solution of your choice. You can store it in the freezer for many years or you can move on to your next experiment. You have just extracted DNA.